This week, we will be looking at several reactions of copper. Sometimes this is called the copper cycle. You will be starting with a copper wire that you will find in your basket, along with a couple of materials and some chemicals. Other chemicals will be found in the hood. It is important that they stay in the hood and that you work with them there because they either give off strong fumes or they are very corrosive and must be handled carefully. In this lab, you'll be working with a pre-cut length of copper wire. We're going to quantitatively look at how much copper we can require after going through all of these reactions. So we want to start with as pure copper as possible. You can see that these two wires are slightly different colors, this one looking more the copper color and this one being a little bit darker. The darker color is copper oxide that has coated the outside. So we don't want to include the oxygen as part of our initial weight as much as we can avoid it. So your first step is going to be to take your sandpaper and to sand the copper wire to get it as shiny as possible. Now you don't need to spend hours and hours on this, just get it as shiny as you can and then move on. Record the mass of the copper wire by using the analytical balance. The mass is very small, so it's important that you use the analytical balance so that you have more than one significant figure. Put your wire in the bottom of a 250 milliliter beaker and take it to the fume hood. Make sure you are wearing gloves, and then you are going to add your 4 milliliters of nitric acid. The exact amount of the acids in the fume hood are not important, just as long as you get close. Be sure that you are ready to record your observations using all of your senses. There will be things to see and hear and smell. Please don't taste. After the copper wire has completely dissolved, add the deionized water. Again, it does not need to be measured precisely. You'll notice that the solution turns a pale blue. You want to make sure that you remain in the hood until all of the fumes are gone, because even once the fumes turn clear, they can be choking and noxious. If at any time you notice your gloves have become contaminated with chemicals, pull them off immediately, inside out, so that the chemicals remain on the inside. It is best not to write in your lab notebook while wearing gloves, because any stray chemicals will end up on your notebook and on your pen. When is waiting for the fumes to disperse in the fume hood, you can be preparing the 30 milliliters of sodium hydroxide at your lab station. Adding acids or bases to water is usually an exothermic reaction, so while stirring, you're going to add your sodium hydroxide. Note the changes in color and clarity in your lab notebook. Take your solution back to the fume hood and gently warm it on the hot plate. You're going to bring it to boiling, so you want to stir it so that we don't have any bumping caused by the beginnings of boiling. Make note of the changes in the solution and in the precipitate before you move on. Recall that the bench tops are not heat-proof, so please set your hot beaker on top of one of the glass plates while you wait for the black precipitate to settle to the bottom. While you wait for the precipitate to settle in the bottom of your beaker, measure out 200 milliliters of deionized water into your 400 milliliter beaker. You can just use the markings on the beaker. You don't need to be very precise with this. And allow this to become very hot. Decant the clear, colorless supernatant fluid off of the top of your precipitate by placing your stirring rod over the top of the glass through the lip of the beaker and pouring out. All of your copper is part of the black precipitate, so make sure that that stays in the beaker. We don't want to lose any of the copper. We also don't want any of the copper to go down the drain. You don't need to completely remove all of the water because you're going to rinse everything with your hot deionized water and repeat this procedure. Once the supernatant fluid again becomes clear, you can decant off this water and move on to the next step. Make sure you are wearing gloves before you work with the sulfuric acid because it is also very corrosive. You don't need to measure exactly 15 milliliters, but get close and transfer it slowly to your remaining black precipitate and stir. You can take the beaker back to your lab station to record your observations. You only need about 0.4 grams of zinc. You don't need the exact amount, so you can weigh it on the top loading balance if those are available to you. If you go a little under 0.4, that works as well. Add the zinc all at once to the beaker and stir. You should begin to notice that it will fizz because it is reacting with the acid, but it will also start to react with the copper in the solution. You should notice the zinc, which is silver colored, is replaced by the brownish color of the copper forming as it is reduced. Once the fizzing has stopped, check your solution. If it appears to still be pale blue in color, you can add a small amount of zinc and continue stirring. If your solution continues to have bits of silver in it, this means there is still zinc present, and we don't want that to be included in the weight with the copper. So you will add a small amount of hydrochloric acid. Transfer your brown copper solid into the pre-weighed evaporating dish. If you haven't weighed your evaporating dish yet, please do so now. You're going to rinse it with a few milliliters of water. Stir it a bit and wait for it to settle, and then decant off the water. Do this a couple of times, and then transfer your evaporating dish to the hood. In the hood, you will add a few milliliters of acetone. Acetone is quite volatile and should not go down the drain. It will also help to clear out the water and evaporate more quickly than the water will. Once you have rinsed your copper precipitate with the acetone, decant into the acetone waste container rather than down the drain. Your copper will still be wet, so you will need to heat it gently on the hot plate. Only do so for a minute or maybe two, because if it stays too long, then it will begin to oxidize.
Wait for your sample to cool and then weigh it on the analytical balance.
Check with your instructor about what to do with your copper sample. They may want you to collect it into a vial or simply have everybody put it into the same waste container. If you place it in the vial, you will need to make sure you have a label that includes your name, the date, and identifying the sample.
Clean and dry your evaporating dish and return it to the basket along with the other items for the next class to use.